Hello everyone. Myself Dr. Vinayak, Assistant Professor in the HOD Department of Botany, SVS College, Bantwal. In today's seminar, I will present my topic on the distribution patterns of the lichens biota in the Shimoga district of Karnataka. Just going to the abstract, this is the presentation of my abstract of the topic. And just first going to the introduction of the topic, the lichens are one of the interesting group of the organisms. Generally they are the specialized group because the only symbiotic association of the algae and the fungi in the nature, these lichens will be formed. So usually generally the lichens are defined as the symbiotic association of a photosynthetic partner and the mycobiont. That's referred as lichens. They are represented almost 85% of the earth surfaces from the tropical to the tundra region. They are all places they distributed. And in India there are about more than 2600 species of lichens are reported, and in the world around 21,000 of the lichens are reported from the world, and especially in the Western Ghats around 950 species of the lichens are reported. These lichens are divided into different types based on the study form, that may be Crustofolios fruticose type, and even they are distributed into corticolous, terricolous, saxicolous lichens based on the substrate form. So these are the different types of the lichens. So coming to the objectives of my study, to make an inventory of the macrolichens in the Shimoga district, and to study the distribution pattern, and to study the substrate ecology of the lichens. So going to the material method, so we have collected the lichens from the different localities of the study area. During the study we have recorded the field localities, temperature, humidity, all the things, ecological parameters we have recorded. So this is a map showing our study areas. This is Shimoga district in the location. So we have mentioned the localities in the study area, and these are the different types of forest, that's evergreen, semi-evergreen, deciduous forest, and the shola forest currently occur in the Shimoga district. And this is the rainfall pattern and the temperature pattern along the Shimoga district in the last 10 years. And going to the methodology, so when we collected the lichens from the field, so we have go through the identification, that's the anatomical and chemical, and the TLC used for the identification. So color test is one of the basic things to identify the lichens, and whatever the collected material that we have stored in the field. This is how the actual anatomical studies we will be identify the algar and the fungal partner in the studies, and we have also calculated different statistical tool to correlate the data. So going to the result, there are about 91 species of the lichens from 31 genera and 14 families that were reported from the study areas. In the study areas, the common species are the Derineria, Fixin, Lechinora, Heteroderma. So these are some common species occurs in the study area, and among that, the substrate form, the mainly majority of the lichens are reported from the corticolous type. This the Venn diagram shows the lichen distribution in the different substrate, that may be corticolous, saxicolous, and the terricolous. The majority of the lichens are reported corticolous. Among the lichen growth form, the mainly the foliose lichens are largely distributed than the fruticose and squamulose type of the lichens, and this is how the showing the different minor substrate, that may be a stem, twig on the lichen distribution. This is the pie chart is showing. And going to the different types of the lichens occurs in the different types of the forest. Among that, the in the deciduous forest you can find commonly the Ramalina, Heteroderma, and Usnea, and Paroderma species. And whatever the reported species, among that 38 species are reported from the evergreen, semi-evergreen forest, and 35 species are reported from the semi-evergreen forest. This is an table showing the distribution of the lichens in the different types of the forest, and some of the lichens were occur both in evergreen and the semi-evergreen and deciduous forest. And if you take the lichen support, that's the growth form of the lichens. So generally the bark texture and the bark moisture and also the microclimate will key role in the lichen distribution pattern. So just look at the different species of the lichens grow on the different tree species, that's usually the termaria, ficus, and jujubas. These are supported for the more number of lichens growth, and xylea and tectona will support it for the less number of the lichens lichens growth. This is a table showing the different host species and their bark pH and the moisture content and their colonization we have recorded in the study areas. And if you look at the algal partner in the lichen symbiont, the majority of them are Tribuxia and very less Nostoc and Cytodium were distributed. These are some common algae or the phycobiont involved in the association of the lichen. And this is how the table is showing the different lichen species and there the statistical values, that's the density, abundance, frequency and also importance value index. So here you can find the Parmotrima are largely distributed when compared with the other species, and this is the graph showing the family importance values of the lichens families in the study area. Among that, the Physaceae and Formulaceae largely distributed, that is followed by the Usneaceae and also the other group of the families. And this is how the diversity index, that's a Shannon diversity index, is represented by 4.41. That's indicated that the lichens are largely distributed and rich in diversity, and Simpson is represented by 0.98. So the almost they indicate the abundance of the lichens in the study area. So this is how the actually the some of the common species of the lichens occur, that's the Parmotrima, Heterodermia and Leptogium and other species. They are commonly grow on the different state. These are all the common the lichens occur in the Shumaga district, and these are the some Telostachys, Usnea, Heterodermia and Leptogium, so etc. And this is the table is showing the checklist of the lichens occur in the study areas and their substrate and also the specificity. These are some common reference we have followed for the studies, and I am very thankful to my supervisor and also the Upriti and Sanju Naik for help in the during the identification of the lichens. I am especially thankful for the organizers to give me an opportunity to talk on this occasion. Once again, I am very thankful all of you to listen my presentation. Thank you. I hope if you have any queries, please ask during the session. Thank you all.